Hello. Today we're going to talk to you about Huntington's disease. So a little background on Huntington's. It's a neurodegenerative disease and it's caused on your fourth chromosome, and it's because of a CAG trinucleotide expansion. Now it's similar to Alzheimer's in the form that it is neurodegenerative and you can see dementia form, but it's also very different because Alzheimer's is in the APP gene, and which has instructions for making amyloid beta precursor proteins, but Huntington's is actually in, the problem comes from CAG trinucleotides that code for the amino acid glutamine. Some symptoms of Huntington's, you usually don't see them until you're between 35 or 45, but sometimes you'll see them before that. The first symptom is chorea, which is like an uncontrollable jerky movement. And as time goes on, you'll see like cognitive abilities affected. This includes like abstract thought, planning, cognitive flexibility, initiation of appropriate responses and actions, and the inhibition of inappropriate actions and responses. After that, you'll start to see memory deficits such as long-term, short-term, episodic, which is episodes of your life, procedural, such as how to, tie your shoe or anything like that, and then working. And ultimately it leads to dementia. So we're going to start with DNA replication, but we're going to start with the key terms you should know. DNA polymerase is an enzyme that carries out DNA replication by adding nucleotides in the 5' to 3' direction. So because of this, when DNA helicase unzips the DNA strand, it makes two daughter strands, and they're single-stranded DNA, and on one strand polymerase can just slide down it from 5' to 3', but on the other strand it has to go the opposite direction in little segments called Okazaki fragments. And we'll get to that later. Another thing you should know is nucleotide pairing. So thymine goes to adenine and cytosine goes to guanine. And an important note for later is that adenine to adenine are very repulsive, whereas thymine to thymine are less repulsive, and this just has to do with the size of thymine and adenine. So here's a diagram of DNA replication. So you see helicase unzips the DNA, causing two single strands. And on the 3' one, polymerase just zips down in one direction without a problem, but on the other strand, it starts at the 5', so it has to go down, RNA primer lays down, and an Okazaki fragment has to be made, and eventually they'll connect to each other. And this is just a diagram of how nucleotides pair up. So you'll see that you have the two strands split into single strands, and then they get connected with their nucleotide. So you'll see C goes to G, A to T, and so on.
Hi, okay, so I'm going to talk to you about the molecular mechanisms of HD, or Huntington's disease. So first I'm going to go over CAG repeats and what they are, kind of. And then I'm going to talk about the intergenerational transmission between parent and offspring with the CAG repeats. And then I will talk about the problems that are caused by these repeats, or just give you a little general overview of them. Alright, so first off, in people without Huntington's disease, they have between 0 to 35 CAG repeats. But within the people who have Huntington's disease, you'd usually find over 40 repeats. Between that, like 36 to 39, it's undetermined. So you're not going to really know for sure if they have the disease or not. Okay, so basically, during DNA replication, helicase comes in, pull apart the strands, and then DNA polymerase comes in and find complementary bases to the template strand. When it reaches these CAG and CTG repeats, DNA polymerase trips, and researchers are still trying to figure out why they trip. But all we know right now is that they trip, and then they add more CAG repeats onto the daughter strand than those that were originally on the original strand. So on the daughter strand, the extra CAG repeats are going to form a hairpin, kind of like this one. And then in the future or the next replication, DNA polymerase might be able to smooth out the CAG repeat hairpin, causing there to be a longer chain of CAG repeats. So that's how expansion occurs. And also, if the hairpin, CAG hairpin, occurs on the daughter strand, there's usually expansion, and if the hairpin occurs on the template strand, there's usually contraction. And contraction is really rare, so. There is a group, a US-Venezuela collaborative research group, and they studied the intergenerational transmission of CAG repeats between parents and their offspring. What they found was that the CAG repeat length depends on the sex of the transmitting parent. So if you are a affected mother, your children's CAG repeat lengths would either not differ that much from yours, or there might even be contraction. But if you are a father and you are affected, your children might have expanded CAG repeats. So that is the finding. And also with if you are a female mother and transmitting it to your offspring, if your offspring is a male, there's more chances that he will have expanded CAG repeats versus if the offspring was female. Okay. And then some problems that are caused by these repeat expansions. During replication, there might be stalling in the replication, because if they can pick up that there's like expanded repeats, then they might stop the replication, fix it somehow, and then continue with the replication. And then the other problem, which we'll go into later, is the additional CAG repeats will add more glutamines to the protein strand, which will cause misfolding because it will end up being bigger, and then that would lead to protein aggregation problems.
Okay, now I'm going to talk to you about protein synthesis and how it relates to Huntington disease. Protein biosynthesis is necessary for every cell's life. Protein biosynthesis is a process where cells generate new proteins. Now we can break this down into two different topics. Transcription, which is the first step of protein synthesis, where DNA is copied to RNA from RNA polymerase. Then translation, which is the process which ribosomes create new proteins. Now, to help you understand the processes, here's the steps to how protein is made. The first step is the DNA unzipped. DNA unzips in the nucleus of the interphase. Then mRNA transcribes it, which mRNA nucleotides transcribe the complementary DNA message. mRNA then leaves the nucleus and goes to the ribosome and reads the codon. tRNA then comes with the anticodon UAC, brings in the proper amino acid from the cytoplasm. Ribosomes add amino acids to the first amino acid with a peptide bond. Then the ribosome then moves down to the next codon for translation. tRNAs are released and bring back amino acids to the growing polypeptide chain. This keeps going until the codon is complete. This diagram shows simply all the steps just as stated. First, it starts with DNA, then it's transcribed to mRNA. Then the mRNA is translated with the attachment of a ribosome, and the tRNA bringing over the right amino acid to produce a polypeptide chain. Now that you understand the process of protein biosynthesis, we're getting into protein folding. Protein folding is the most important thing to know when dealing with Huntington disease. Functional shape. As a polypeptide chain is made for the ribosome, its goal is to form a 3D structure so that it's long-lasting. As it is growing on the ribosome, its peptide is folding over, which is called functional shape. Native state, don't get confused, is the end structure of the protein. It is this final 3D structure that is stable and functional. Hydrophobic effect. This helps determine the shape of the protein. The proteins have a tendency for hydrophobic, oily portions of the protein to shield the cell from the hydrophilic interior of the cell by burying it into the interior of the protein. Why protein folding is important. The shape of the protein is determined by the amino acid sequence. The shape of the protein determines its function and capabilities as a protein. If the protein is not able to reach its native state, which is its final stable shape, it could cause toxic functionality. This is common in a lot of neurodegenerative diseases such as Alzheimer's and Huntington's disease. Right. This slide gives you a visual of the native structure and how it starts. This is a long polypeptide chain, and then it goes into a 3D coil, is like usually what proteins do. That would be its native shape. That's just an example for you. Protein aggregation. The biological phenomenon in which misfolded proteins accumulate and clump together, either inter or extracellular. Some reasons for misfolding that are common are temperature, external fields such as electricity or magnetic fields, molecular crowding, or pH. Why is protein aggregation important when it comes to Huntington's disease? Huntington's disease is in fact due to the misfolding of proteins, which would cause aggregates. Huntington's disease is called an intracytoplasmic aggregation disease, which is a disease where misfolded proteins within a cell aggregate, which causes the cell to die. The gene that has misfolded proteins that are produced in the 4th chromosome in the HD Huntington gene. The CAG nucleotides form a long chain which causes the misfolding.
So now we'll be discussing protein aggregation and its consequences. As mentioned previously, people with Huntington's have an increased number of these CAG repeats in their Huntington gene, coding for an excess number of glutamine in their Huntington protein. This glutamine extension in altered Huntington leads to further problems, mainly protein aggregation, which leads to eventually more serious problems. Some of the topics we will be discussing about protein aggregation will be the actual aggregation of the HD protein, which is the Huntington, the altered ubiquitin proteasome system, the consequences of the HTT aggregates, neural inclusions, and the altered Huntington and CREB binding protein. So because glutamine is polar, or a charged molecule, the overabundance of glutamine causes links to form within and between the proteins. HTT molecules, that's just the name given to the mutated Huntington protein, stick to one another, forming strands that are held together by hydrogen bonds. Rather than folding into functional proteins, they develop into tangled, rigid groupings known as protein aggregates. These fibrous proteins aggregates accumulate and interfere with the normal nerve cell function. So one of the problems caused by the aggregation of HTT is the alteration of the ubiquitin proteasome system. Normal cells have a mechanism for quality control called the ubiquitin proteasome system, UPS. The UPS is a protein chaperone system which tags misfolded or damaged proteins for refolding, or more commonly for degradation. However, the HTT aggregates can inhibit the function of the proteasomes and deprive them from their normal functioning. As a result, the HTT proteins continue to aggregate, as we can see as well in the diagram that compares a normal pathway with the HD pathway, which just means that it becomes like a vicious cycle because the proteasomes are not functioning, so the aggregates continue to expand and continue to aggregate. So some of the consequences of the HTT aggregates. So they interfere with nerve cell function by entrapping key cell regulatory factors. They further disrupt the normal UPS functioning, as I previously mentioned. And the excess glutamine in HTT aggregates are correlated with the occurrence of nuclear inclusions that I'll talk later. And then the altered Huntington inhibits the CREB binding protein, which I'll mention as well, all leading to overall cell toxicity caused by corrupted metabolic pathways. Neural inclusions. The excess glutamines in HTT can lead to a type of protein building known as neural inclusions or inclusion bodies. The neural inclusions initially form at the axons and dendrites of nerve cells in specific areas of the human brain, producing damaged neurons, which is a characteristic of Huntington's. When they move into the actual cell nucleus of the nerve cell, the condition of the actual neuron becomes worse. So some of the consequences that neural inclusions can cause would be change in cell structure, interference with normal production of other proteins that can lead to both death and degradation of neural cells. Eventually, the HD symptoms will become evident after prolonged neural degradation. So now I'm going to talk about the altered Huntington and CREB binding protein. CREB, also called CBP, is a small regulatory protein which is key for cell survival. CBP has also exchange of 18 glutamines which interact directly with the expanded HD protein because it also contains glutamine. Huntington aggregates pull CBP away from its normal position, inactivating its normal functioning. With CBP inactive and malfunctioning, the survival pathways that this protein carried out by inactivating the respective gene can no longer occur, leading to nerve cell death. CBP shortage on the brain of dead HD cases supports this theory. Now, this is just a small recap. So, CAG expansion can trigger mutant HTT, which after prolonged aggregation can cause cytotoxicity and eventually neuronal cell death. This neurodegeneration on the brain cells causes uncontrolled movements, loss of intellectual faculties, and emotional disturbances. The late onset of HD is due to the fact that the aggregation of HTT after substantial alteration of the UPS is gradual, as well as the actual neurodegeneration. Thank you.